Welcome everybody here in real life and also online. It's a special day today for many reasons. Today is Tuesday, so and it's February 22nd in the year 2022. So there's a lot of twos today, and we're celebrating one of our very own. This is a celebration of the work done by our postdocs, and Alcino will introduce the Arnold Scheibel Distinguished Postdoctoral Lecturer, Megha Sehgal, just in a minute. I would just like to say a few words about Arnold Scheibel, who was a very distinguished researcher here at UCLA. He was born in 1923 and unfortunately passed away just a couple of years ago in 2017. He's left a very big legacy at UCLA. He's endowed 3 chairs for researchers. He's also still, his estate is supporting undergraduate education at a really nice rate on a yearly basis. And we hope this will go on for a very long time to come. Arnie he was a MD. He went to medical school at Columbia. His studies were interrupted by the war effort. But then he concluded his studies first at Columbia and then at Mount Sinai, never wanting to really go anywhere above the neck. So he was really interested in cardiology, but then sort of over time saw the light and did a lot of research, mainly anatomy in the both spinal cord or all 3 spinal cord, brainstem, and then venturing out into the cerebellum. He did a lot of really nice connectivity in the brain. So he saw how all of these areas were organized and were talking with each other using a lot of different techniques, including at that time the Golgi stain, which was still very much in vogue. He came to UCLA was hired by Ted Magoon in 1955 and stayed here ever since until his death. He did a lot of teaching at UCLA. He taught undergraduates mainly. And one of the things that I found extraordinary about his teaching was when I talked to some of the undergraduates, they said, oh, we really love him. He's a fantastic teacher. And then I asked, what grade did you get in the class? And they said, oh, I got a D. So they really failed, but they still loved him as a teacher. So it wasn't just that he sort of encouraged people by giving good grades. He really encouraged them by teaching them the material. So it's in his honor that we honor postdocs. And as we're doing that, I call on Alcino that we can give Megha her prize, which is a really nice plaque. Maybe I can officially hand you this. And then we can shake your hand or do something as Lauren takes a picture. Okay. All right. Yes. Oh, that's right. We need to stand within the white spaces here. Very important. Very important. All right. Okay. All right.
Thank you so much.
Thank you very much. And Alcino.
It is such a pleasure to introduce this year's distinguished lecturer, Arnold Scheibel Distinguished Lecturer, Megha Sehgal. Megha is being recognized for her fantastic work in somatic and dendritic mechanisms that link memories across time, as well as for service to our UCLA community. Megha has had a key role in both, and I'll mention that in a second. Megha graduated from the University of Delhi in 2006, for which I'm very envious. What a fantastic, interesting place to have been. And for her PhD, she worked with James Moyer in Wisconsin, where she carried out groundbreaking work, actually beautiful work on the relationship between learning-related intrinsic and synaptic plasticity. Amongst other things, she was one of the first to show that learning in the amygdala triggers changes in intrinsic plasticity and that these changes are coordinated with changes in synaptic plasticity. So we've always known that there should be a relation between the two, and Megha is one of the pioneers in actually demonstrating that relation. She also studied aging and found that age-related deficits begin early in middle age. And now for that, I'm not very happy, actually. And that they are very sub-region specific, very region brain specific, which is also another important contribution. After completing her PhD, our lab was fortunate enough to have Megha joining her. One of her labs is a judgment, but nevertheless, there she is. And she has been working on memory linking, and she will tell you about these really wonderful results in a minute. So besides the fantastic work that you'll hear from her today that she carried out in the lab, she has had a key role in the Miniscope research community at UCLA. Not only has she been an active participant in the development of these tools, she has also had a central role in their dissemination at UCLA and internationally by helping to teach a number of workshops here and abroad, actually. At UCLA, she has also had a key role in the UCLA learning and memory community. She had she was one of the leaders of our journal club, our very successful journal club. So thank you, Megha. And I would be amiss if I didn't say something about her work. I said she joined our lab, but actually that's not exactly true. The truth is that she joined our lab and Josh Streckenberg's lab and Payman Ghoshadi's lab, because she has worked in all three labs, especially in Josh's lab. You'll see a lot of the work that she'll share with you here today was really a very intimate collaboration between our two labs. We could not have done it without Josh. And a lot of the work that she has participated at UCLA could not have been done without Payman. So, UCLA is known for this type of collaborative work, but for it to actually take place, you need people with very high social intelligence, like Megha, people that everybody loves and doesn't mind working with. And, she's really wonderful, generous, creative, and courageous. Well, she did join our lab, so that takes guts to start with. Anyway, okay, without further ado, please join me in congratulating Megha Sehgal, this year's Arnold Scheibel Distinguished Lecturer. Welcome, Megha.
Share my screen. Mm-hmm. Well, thank you so much, Alcino. That was a very kind and generous introduction, and I am very, very honored. And a lot of these things were completely my pleasure. I had so much fun working at UCLA that, there's never a time where I don't recommend that people come here and work here. So with that, I wanted to start talking about the work that we're here to talk about. And specifically, what I'm going to talk to you about today, how our brain links to memories. And with these, in order for these memories to be linked, how there are somatic mechanisms or neuronal mechanisms that are necessary. And in addition, what's new here is we also have these branch-specific or highly compartmentalized dendritic mechanisms that are necessary for two memories to become linked. And this is sort of the take-home message of the talk. So intuitively, we all realize that memories or single memories don't occur in isolation. They often interact with one another. In fact, our brain organizes them into a framework so that we can pull from this rich framework and make adaptive decisions. So I'll give you one example. This is one of my favorite memories. This is my partner. We took a trip a few years ago to Hawaii, and this was one of our favorite trips. We started the day by enjoying sunrise on one side of the big island. We traveled across the island on the same day and ended up on the other side and enjoyed a sunset. So this was a great vacation, and we hoped for a redo, and we went back to Hawaii a few years later. When we did this, Mother Nature was not so kind. It rained the entire trip. We only managed to get out of the hotel room once, and that is to rent ATVs and get muddy. So I tell you this example because not only is it one of my favorite memories, well, half of it is a favorite memory that I like to reminisce about, but also because it highlights how our brain organizes memories. So we organize memories together because they might happen on the same day, like the first two pictures. We might organize them together because they happen in the same place, like these three pictures were taken in Hawaii. Or we might organize them together because they are either good or bad memories. So the first two are a memory of a great vacation. The other one is not such a great vacation. So our lab is interested in understanding how these memories are linked together and what are the different dimensions. Specifically, today I'm going to talk to you about how our brain links memories if they happen close in time. So if two memories are acquired within hours, we link them and how this happens. And the reason we're so interested in this aspect is because you can imagine that linking memories that happen close in time allows us to make an episodic timeline of events. It has predictive value that we can draw from. In addition, we also hope that we uncover the mechanism by which memories that are temporally proximate are linked. We also might understand the mechanism the brain uses for organizing memories across other dimensions. So with that, what do we understand about the neural mechanisms of this? We know that single memories are encoded in sparse, but a distributed network, sparse population of neurons in a distributed network of brain regions. We know that certain allocation mechanisms at the neuronal level decide which neurons are going to be allocated to a particular memory. For example, a lot of work from our lab and other labs has shown that if you increase the level of Krebs, these neurons are now more likely to be recruited into a memory. We also know that this change in Krebs changes intrinsic excitability or the excitability of the neuron, which predisposes it to be recruited into another memory. We also know, as Alcino hinted, some of my work also shows that once a neuronal ensemble is involved in learning, it changes intrinsic excitability itself. So putting these two pieces of information, Alcino had a hypothesis where we said we can use these allocation mechanisms to link memories. So for example, since we have a set of neurons that are undergoing learning, they change their levels of Krebs, they also change intrinsic excitability. Now these neurons are for a time being more likely to be recruited for a memory. And this is what we see. If another memory is acquired within a particular amount of time, then some of the same neurons are recruited to encode this memory. Hence, these two memories are recruited by overlapping neuronal ensembles. We think because of this overlap, a link between these memories can be formed. For example, since these two memories are encoded by some of these overlapping neurons, when you recall one memory, you can also recall another. Hence, our brain helps us link these memories. So I have to give a shout out to Denise. She was a postdoc in our lab who published a seminal paper showing exactly this. So what she did was she had mice, which are our model system, and she had mice explore 3 different contexts. These contexts were either explored within the same day or 5 hours apart, or they were explored 7 days apart. She found that when 2 contexts are explored 5 hours or within the same day, they had more of an overlap in the neural ensemble that encoded them. But the ensemble overlap dramatically decreased when the contexts are explored 7 days apart. And she showed this in hippocampal CA1 brain region. She did not stop there. She extended this behaviorally. What she showed is, so if you see here, these 3 contexts remain the same. Mice see them either 7 days or 5 hours apart. But then she went ahead and paired one of these contexts with a foot shock. Obviously, the mice are afraid of this context. And if you test them, like in the first bar here, in this context, they freeze. Of course, it's a scary context. Surprisingly, if she tested the mice in a context that they had seen, again, earlier in the day, but were never shocked in, they also freeze in that context. Finally, she tested them either in a novel context or a context that they'd seen 7 days before. And what she found was that this transfer of fear to a context that they'd seen earlier in the day is specific. The mice don't do this for another context they'd seen a week before or a context that they'd never seen, indicating that indeed, two memories that are acquired close in time are not only encoded by the same neurons, they also behaviorally linked. So when you assign a scary memory to one of them, the other one also becomes scary. However, so far, we've only invoked neuronal mechanisms. A neuron does not look like this cartoon drawing. It looks more like this. And our idea was that we understand that there are neuronal mechanisms, but what happens in the rest of the neuronal body? There's a lot of real estate dedicated to these dendrites. Are they undergoing any plasticity that contributes to this process? And that is sort of the theme of the talk today. Indeed, there is a lot of literature going back from early 2000s that shows that indeed dendritic compartments undergo localized compartment-specific plasticity. And although many different mechanisms have been invoked for this, a common theme is that most of this plasticity affects future plasticity within the same dendritic compartments. And this is what we wanted to test. And in order to do this, we not only looked at the neuronal compartments, while the animals encoded two memories close in time, we also tracked which dendritic branches were encoding the first memory. And then we looked a little bit deeper and saw what were the spine changes on these dendritic branches. And we followed this up by looking at a second memory that was linked because it was encoded very close in time. We did all of this in retrosplenial cortex, and there are several reasons I use this brain area. So retrosplenial cortex in recent years has gotten a lot of attention, and one of the reasons is it's involved in a variety of behavioral tasks. More important or pertinent for today's talk, it's involved in spatial and contextual learning. We also know some of the work from our lab and other labs, what the neural correlates as well as the dendritic plasticity correlates of contextual memory formation in retrosplenial are. And then finally, I think one of the main reasons it's gotten so much attention is, as you can appreciate here, it's a midline dorsal cortical structure. And this makes it very easily accessible to longitudinal imaging techniques that can be very invasive in some other brain areas. However, we did not know what happens in retrosplenial cortex during contextual memory linking. So that was the first question we addressed. And to do this, we used Miniscope imaging. Miniscopes, specifically, we used UCLA Miniscopes that have been developed here as a collaboration between our own lab, Peyman Gulshani lab, Silva lab, and Baljit Cox lab. And for those of you who may not know, these are miniature microscopes that are open source and highly customizable. You can take the hardware and software and adapt it for your purpose. More importantly, for us, these miniscopes are small enough that our lab heroes, mice, can easily wear them on their head, and we can have them do various tasks. So using this, we injected GCAM and adapted the system to be able to image calcium transients within the RSC. And this is just an example video which shows you raw calcium imaging data as we see it from a miniscope in RSC, and this is just a lightly processed video. So using these miniscopes, we've done various different tasks. Specifically, here you see a mouse running on a linear track back and forth while we isolate neuronal segments as extracted from CNMFE. And now we can correlate behavior with these extracted segments. So using the system, we first addressed, is there any co-allocation or is there any overlap in the neuronal ensemble that encodes linked memories within RSC? We injected GCAM6F into the retrosplenial cortex, implanted a grin lens on top of it, and then used a miniscope to image. And if you're interested in where our GCAM expression is located, this image tells you that too. And this is a CNMF extracted example video. And as you can appreciate, not only can we extract segments or neurons from these calcium videos, we can also extract the calcium dynamics of these neurons. With that, we asked if we do the same memory linking task or the mice explore 3 contexts, either separated by 5 hours or 7 days, what happens to neuronal ensemble within the RSE that's encoding this memory. And just to point out, we're going to use a very similar imaging paradigm for the rest of the talk. So when I do this, I realize that the overlapping segments, which you can see from an example mouse here, are much more, or there are many more overlapping neurons or neurons that are recruited for both tasks when the contexts are explored within 5 hours of each other. And this number dramatically reduces when the mouse sees the two contexts 7 days apart. You can also see this is true at a broader level where most mice increase the overlap when contexts are seen within the same day. So this was great. This told us that yes, not just in hippocampal CA1 neurons, but also in retrosplenial cortex neurons, we see an increased overlap if contexts are acquired within the same day. We went ahead and made sure that this is not because of any changes in imaging conditions. We gave mice either the same context or two different contexts 7 days apart. And we find that indeed, when the mice see the same context, they see same context 7 days apart, there's greater neuronal overlap that's recruited to encode these contexts in comparison to contexts that are different from each other. This data that I'm not presenting from our lab, we've also gone ahead and looked at the neuronal activity dynamics of these retrosplenial cortex neurons. And we find that whether we look at activity or co-activity patterns, both of them tend to remain similar when the contexts are seen within the same day versus 7 days apart. So this is great. Just like we found out that just like hippocampal CA1 neurons, retrosplenial neurons also encode two contexts that are linked with overlapping neurons. What this does not tell us at all is if this overlap is in any way functionally relevant. Just because we see something doesn't mean it's mediating behavior. So that was the next question we tackled. And to do this, we used the TED tag system that was first developed in Mark Mayford's lab. The TED tag system briefly consists of two genetic elements. The first genetic element consists of a CFOS promoter. The CFOS promoter, or CFOS, is an immediate early gene and has been widely used to be an activity-dependent marker and to tag neurons or study neurons that are involved in a particular behavior. The second element basically senses this CFOS activity and makes an effector protein. In our case, this effector protein is going to be channel rhodopsin, and it allows us to later on optogenetically activate these cells. The beauty of the system also is that this is under the control of doxycycline. So the mice can be on a doxycycline feed and we can turn the system on and off. To illustrate this, this is an example of an RSE brain slice or retrosplenial brain slice when the mouse is on doxycycline. There's absolutely no expression. When you take the mouse off doxycycline, then you can turn the system on and now you can tag the neurons that are important for the relevant behavior. So we asked a very simple question first. Can we move behavior by manipulating neurons in RSE or retrosplenial cortex? We took the mice, we put them in a box, we shocked the mice in this box. So this context is now fearful. We also took the mice off doxycycline while we did this. So we tagged every neuron that was CFOS positive, or most neurons that were CFOS positive in retrosplenial using this. Next day, we bring the mice back and we put them in a novel context. And we optogenetically reactivate the cells that were tagged a day before. I do want to point out a very similar study had been done. We just wanted to make sure that in our system, in our hands, this is indeed functional. And what we find is that indeed, while the control mice do not change how they behave or how they freeze in response to optogenetic stimulation, the TED tag mice increase their freezing in response to a baseline period, indicating that we can tag neurons in retrosplenial cortex, we can activate them, and now we can make the mice freeze where they would have otherwise been exploring the context. Obviously, the question that we were really interested in is, this is great, this happens when you look at a single memory. What happens when you take neurons that are encoding a linked memory? Can you reactivate these neurons and can you see behavioral expression change? So this is what we do next. We again put the mouse in a new context. We tag all the neurons that are activated in this context. We take the mice and we put the mice back on dox. And then 5 hours later, the mice see another context. And this context now is paired with a foot shock. So this context is scary to the mice. Again, just like before, the next day, we bring the mice back in a novel context that they're not afraid of. We optogenetically reactivate the neurons in the RSC that were tagged following the first context exploration. When we do this again, control mice don't change their behavior, but the TED tag mice increase freezing in response to optogenetic activation of these cells. So I remind you, these cells are tagged in a context that was never actually paired with a foot shock. In addition, a subset of these animals to just being put back on high doxycycline chow, we also injected that doxycycline interpredatorinally to make sure that we are actually shutting up the system. And we're only specifically tagging the first context. So what do we think is happening? We think that when we put the mice in the first context, we tag the orange cells or cells within the RSC that are responsible for encoding this context. 5 hours later, because of memory allocation mechanisms, some of the same cells are recruited. The next day, when we optogenetically reactivate the orange cells, this is indeed sufficient to trigger the activation of the rest of the circuit that represents the blue circuit, and hence the memory of the context B, which was scary, and hence the mice freeze. At least this is our working hypothesis. And importantly, all of this is done, or this manipulation was done in retrosplenial cortex. So just retrosplenial cortex is enough to manipulate this behavior. So this was great, but we asked a slightly different question next, because this doesn't really address whether neuronal overlap itself is necessary. I do want to point out before we go there that we also tested all of these mice again to assess whether they actually link these two memories. And there weren't any differences because of actually learning the context or linking the memories. So we bring these mice back and we test them in the original two contexts that they've seen, the shocked and the linked context, and we tested them in yet another novel context. When we do this, both groups of mice show similar freezing in the context A, which was the linked context, and context B, which is the context in which they were shocked, and they don't freeze in a novel context that they'd never seen. And another reason I bring this or I point this out is because this is tends to be a really complicated paradigm where mice see a lot of different boxes and freeze in some and don't freeze in another. But typically in lab, when we know memory linking has worked, we see this characteristic V shape pattern where mice freeze in the first context, don't freeze in the next one, and then freeze again. So anytime we see this V pattern for the rest of the talk, we can say that mice linked or did not link. So with that, we were ready to ask our next question, which was that can we change neuronal overlap just within the RSC, and can we now link two memories that are otherwise independent of one another? Again, we use the TED tag system. And we made sure that our mice saw two contexts that were separated by 2 days. And the reason we chose 2 days is that we know when two contexts are seen 2 days apart, but not 1 day apart, these two memories are now independent. At 1 day, they will still link memories. And we know this also because, again, we can shock them in one of the contexts and then test them in a novel context in addition to the previously seen context. When we do this, we find that obviously the mice freeze in the blue context because they were shocked in it. But this freezing is much more than what you would expect in a novel context that they had never seen, or a novel but neutral context that they had seen 2 days before. So with this, we did our actual manipulation. We took the TED tag mice and we exposed them to a novel context. We tagged all the neurons that were activated during this contextual exposure. Next day, in their home cage, we reactivate these neurons. And I've shown you before that reactivating these neurons reactivates the fear memory or the contextual memory. And then a day later, mice see another context. So typically in this situation, mice don't link memories. But now, if we test the mice after optogenetic reactivation, they actually link these two memories. So while the control mice only freeze in the shocked context, mice that had their retrosplenial cortical neurons reactivated on the intervening day are able to link these two memories. They freeze at high levels in the previously seen context, as well as the shocked context, but not in a neutral context that they'd never seen. This indicated to us that not only can we reactivate linked memory and reactivate fear, we can also manipulate the overlap within retrosplenial cortex and now manipulate the behavior of the animals and specifically extend the window for memory linking. We wanted to double check that this is not just something that happens only with optogenetics. So we did pretty much a similar experiment, but now we use DREADS or chemogenetics, and we found the same thing. So this phenomena is pretty robust. And so with that, I wanted to summarize what I've shown you so far, which is that we used miniscopes to show that the same or as overlapping retrosplenial cortical ensemble was recruited to encode two memories that are linked. And we can behaviorally manipulate, or we can optogenetically manipulate the ensembles within RSC. And if you reactivate the linked memory, you reactivate a memory you also activate a memory that's linked to it. And you can move this overlap and move linking between two memories. But I started off talking to you about dendrites. So let's shift our focus and talk about dendrites. This basically allowed us to assess that RSC is a good place where we can look at dendritic mechanisms, because indeed, memories are manipulated, or contextual memory linking can be manipulated by using the retrosplenial cortex. So initially, I talked about how we think intrinsic excitability change is a mechanism by which these neuronal co-allocation or allocation is taking place. For those of you who have never really thought about excitability, intrinsic excitability change refers to a change in either the number of ion channels or the function of ion channels or the distribution of ion channels such that the output of the neuron is modulated. Obviously, this change can be localized to soma or the perisomatic regions. And in this case, the output of the entire neuron is going to be changed. Also, something different is possible. It's possible that we change ion channel distribution or functionality at small stretches of dendrites. And when this happens, the output of the neuron is changed in a more nuanced way. We can turn the output of the neuron on or up only for certain dendritic compartments. And we wondered whether that was happening. Looking back at the literature, as early as 2004, Dan Johnson's lab had shown that indeed localized dendritic plasticity is possible. They used calcium imaging and paired it with LTP induction. And when they induced LTP at certain dendritic branches, they found that back propagation of this action potential was facilitated near the site of stimulation, but not even closer to the soma, but away from the site of stimulation, indicating that there is indeed localized dendritic plasticity within these neurons. However, even though we know that there's dendritic plasticity, the exact role of this dendritic plasticity and how it modulates learning and behavior is very poorly understood. And this is what we sort of sought to address. Our hypothesis was that most of these localized changes in dendritic plasticity make future plasticity much more easier within the same compartments. So if we looked at these dendritic compartments that were involved in the first memory formation, we expected that there might be gradients of locally enhanced intrinsic excitability. They might also include protein synthesis changes. And the net result of this gradient of locally enhanced excitability is that a second memory that's formed within a particular time period is going to be co-allocated to the same dendritic branches. So we sought to address this by looking at dendritic calcium transients. And obviously, we cannot use miniscopes yet for that. So as Alcino mentioned, we collaborated with Josh's lab. And Josh is the most generous collaborator. Thank you, Josh. So all of this work was done in his lab. Again, we used GCAM6F mediated calcium transients within the RSC apical dendrites, primarily originating from layer 5. We used a 2-photon microscope to image these transients longitudinally. And we did this exactly using a very similar setup. So now, instead of mice exploring 3 different contexts, mice are exposed to 3 different contexts in a head fixed setting. Again, these context exposures are either separated by 7 days or 5 hours. We can not only isolate calcium transients within the apical dendrites, we can also look at the calcium output of these dendritic transients. And more importantly, we can track the same dendritic branches across days when we do this. Again, we asked, is there any neuronal or is there any dendritic co-allocation in this behavioral paradigm? So this is data from one mouse. And you can see here that the number of overlapping dendritic segments that were seen, or dendritic segments that were reactivated when the mouse sees another context, is much more when the distance between these contexts is 5 hours versus if these contexts are seen 7 days apart. This is data from one mouse, but we found this to be the case in every single mouse that we looked at. So this is a really robust phenomena. And importantly, we again did the same control. We wanted to make sure that this is not just happening because our imaging conditions are changing. And when we do this and the mice see the same context 7 days apart, there is a lot more dendritic overlap in this condition than when mice see 2 different contexts 7 days apart. So this was great. We are seeing first hints of dendritic mechanisms at play when we're linking 2 memories. So just to again summarize, we've seen that not only are neuronal allocation mechanisms involved in recruiting overlapping neuronal neurons within the RSC, we also see that 2-photon dendritic calcium imaging shows us that we're also recruiting overlapping dendritic segments. So our lab is a big believer in using complementary approaches to ask same questions. Every technique has a caveat, and if you want to get away from it, use something that has different limitations and different strengths. So this is what we did. We basically looked at dendritic segments and assessed spine changes on these dendritic segments. So in order to do this, we used Taiwan YFP mice. These mice label RSC and other cortical neurons in a really sparse way. This allows us to track these dendritic segments across days and see how spines are being added or eliminated from these dendritic segments. In addition, we can also look if these segments or these spines are being added close to each other or further apart from each other. And again, we use a very similar behavioral paradigm. Mice again see 3 contexts. Either they're separated by 5 hours or 7 days. In addition, now 2 hours after every single context exposure, we bring the mice in and we assess spine changes on these dendrites. This is also preceded by 2 days of baseline imaging, so we know how spine dynamics change without behavioral manipulation. So using this system, the first question we asked was, do we see any bias in how single spines are being added to dendritic segments? Are they being added to the same dendritic segments or different dendritic segments? And we did this by simply correlating the number of spines that were added following each contextual exposure. What we find is that when 2 contexts are linked or seen close together, such that the recall of one leads to the recall of other, the spine addition is correlated. That is, the same dendritic segments are gaining spines when contexts are seen close in time. So the second question we asked was, what happens to clustered spine addition? And the reason we looked at clustered spine addition is because clustered plasticity is a hallmark of contextual learning within RSC, and clustered spines are also known to have a disproportionate response for the cell's tuning properties. To do this, we used a slightly different approach. We took any dendrite that had a clustered spine, added to it following one context exposure, and we shuffled the clustered spines as a result of the second context exposure. What this allows us to see is what would happen if spines or clustered spines were being added randomly. And if the spines were indeed added randomly, we would see a distribution like the gray bars that we see here. And when mice see 2 contexts 7 days apart, indeed, the distribution of clustered spine addition is random. You can see it lies right in the middle of this histogram. However, when mice see 2 contexts close in time, such that the recall of one is going to trigger the recall of other, we see that the observed value of co-clustering is much higher than what you would expect by chance. So indeed, not only are the same dendritic segments being activated, the same dendritic segments are also undergoing synaptic plasticity when memories are formed that are linked. And this is all happening in retrosplenial cortex. I do want to point out that both the clustered spine effect as well as the single spine addition effect being biased to the same dendrites is only seen when contexts are explored close in time or within the same day. We also did controls where mice just saw two contexts either separated by 7 days, or the mice were imaged at the same time periods, but we never exposed them to a context. And we don't see any of these effects. So this effect is pretty specific to context exposure and spine dynamics following context exposures. So again, we show you that within the retrosplenial cortex, neuronal co-allocation is triggered by linking of contextual memories, and we can manipulate this neuronal co-allocation and manipulate linking of memories. We also show you using two imaging techniques that not only are the same dendrites activated, they also undergo synaptic plasticity on overlapping dendritic branches when contextual memories are formed close in time. So this led us to a point where we looked at neuronal co-allocation using two different methods. And this is sort of a hallmark of what I've learned at UCLA, in Alcino's lab, and at ICLM. If you want to look at phenomena that's important for behavior, you observe it. And then when you can, you manipulate it. So we observed the neuronal allocation mechanisms in RSC using UCLA Miniscopes. We used the TED-TAG system to manipulate it. And this gave us a pretty reasonable confidence that this system is actually, our neuronal allocation mechanisms in RSC are indeed important. When we started looking at dendritic allocation mechanisms, we were able to observe it using two complementary approaches, looking at either the calcium transients within dendrites or looking at synaptic plasticity or spine changes within RSC dendrites. We also wanted to manipulate it, but this is where we sort of hit a wall. There were really no good ways to manipulate dendritic plasticity mechanisms. In fact, a lot of the previous studies are mostly observational. We haven't really been able to find ways to manipulate dendritic function. So we got lucky because as I was starting this project, a paper from Hirao Kasai's lab just came out. And this really inspired us, and we collaborated with Wando Ho to make a slightly different version of this construct. Briefly, what Hirao Kasai's lab does is they used Ph.D. 95 and combined it with DTE, which is the dendritic targeting element of an ARC mRNA. And when you use these two things together, you can now shuttle any photoactivatable molecule to previously activated synapses. We were interested in dendrites, so we just used the DTE part of this construct. And I think we used it in a way that makes it slightly better. So we put this entire DTE construct, fused it with channel rhodopsin, and we put it under the TED-TAG system. Now we have two ways to control activity. The TED-TAG system means that we can control it using DOCS, and it's only turned on in neurons that are important for the activity or are known to be important for behavior. In addition, channel rhodopsin is fused to the dendritic targeting element, and it only goes to the dendritic compartments that are recently activated. And we think this works pretty well because when we look at channel rhodopsin expression without DTE in a TED-TAG system, you can see it's enriched both in neuronal cell bodies as well as dendrites. But with fusion with DTE, it's expressed sparsely only in dendritic compartments. So this told us that indeed, we're seeing labeling of dendritic compartments, but is the functionality also restricted to dendritic compartments? For example, if we activate these dendrites, is the activation limited to the dendrites? So I've shown you this data before where we tag neurons within the RSC and we reactivate the fear memory. Now we did a slightly different experiment. We asked, can we tag the dendrites and can we reactivate the dendrites, and what happens to the fear memory? So when we do this, we find that just tagging the dendrites and reactivating them in a new context was not sufficient for the expression of fear memory, which indicated to us that not only are we tagging these dendrites, we can reactivate them, but at least the activation of these dendrites is not sufficient to activate soma. And hence, we can look at it as a specific marker for dendritic reactivation and dendritic plasticity. Using this system, we asked, again, a very similar question that we had before. Can we now, instead of manipulating neuronal overlap, manipulate dendritic overlap in RSC? And when we do this, can we change the window during which memories are linked together? So we bring the animals back, put them in a new context, and take them off docs to tag any dendrites that were activated during this exploration. Animals come back and we reactivate just those dendrites optogenetically. Mice see another context 2 days later. So typically, I remind you, these contexts are not linked, but now we're bringing some animals back where the dendrites are reactivated. And one of these contexts is paired with a shock, and we test them in 3 different contexts. Just like before, our control animals don't show memory linking. They freeze similarly in a context they'd seen before, 2 days before, and a novel context, but freeze at high levels in the shocked context. However, if you tag the dendrites and reactivate them a day later, now you see the characteristic V pattern, or the animals link these 2 memories. They freeze at similar levels in both the contexts that they'd seen previously, even though they were 2 days apart. And this freezing is much higher than another novel context that they'd never seen, indicating to us that not only can we manipulate neuronal overlap, we can also manipulate dendritic overlap just within the RSC and manipulate linking of memories, contextual memories. So this gave us a good idea that dendritic plasticity is indeed sufficient for manipulating memory linking. We also wanted to see how all of this plays together, how neuronal and dendritic excitability acts together to manipulate linking of memories. So here we're fortunate that Alcino has a long-term collaboration with Yota Poirazi, and she was very happy to help us with these modeling work. So she has an existing model that predicts how neuronal inputs are used and dendritic and somatic changes bring about linking of 2 memories. So, and she's published with this model before. We made certain tweaks to this model. Specifically, the linking model consists of 2 plasticity mechanisms. One of them is a somatic excitability change, and the second component is a change in dendritic excitability. And there's a rule where every time the potentiation of spines is more likely to happen if there are other potentiated spines on these dendritic branches, so an allocation mechanism. We wanted to see whether with a linking model that has dendritic component and no dendritic plasticity component, is the linking of the 2 memories different? And there's various aspects to this model, but the output of the model is during recall. So during recall, we find that if the linking model has both dendritic and somatic excitability elements, the co-recall of memory is facilitated when the memories are encoded 5 hours apart or within the same day. And this co-recall reduces as the memories go further apart. However, if you take out the dendritic excitability and allocation mechanisms from this model, you do not see any effective time. Memories 5 hours or 2 days or 7 days apart are co-recalled at same levels, which is much lower than what you would expect when dendritic mechanisms are in play. So this predicts to us that not only is it sufficient to have dendritic plasticity mechanism for linking of memory, it might also be necessary. Okay, so to summarize, I've shown you data using Miniscopes and the TED-TAG system that neuronal co-allocation plays a critical role in linking of 2 contextual memories. I've also shown you 2 sets of imaging data that shows that not only are the same dendrites activated, they're also undergoing synaptic plasticity in an overlapping manner to recruit 2 linked memories or to encode 2 linked memories. And then with modeling and optogenetic manipulation, with modeling work and optogenetic manipulation, we try to show that this mechanism is both necessary as well as sufficient. And what we really think is happening is there is synergism between somatic and dendritic plasticity changes that allow us to link memories together. And since a lot of learning and memory studies sort of ignore what's happening at dendritic compartments, this sort of brings attention to the fact that we should be looking at dendrites a lot more. I also think that this brings up a lot of interesting implications. So for example, there's been a recent paper, and by recent, I mean a week ago, from Mac Harnett's lab, where they track the inputs for RSE layer 5 neurons and where these dendritic inputs are coming from. This, along with other work in cortical neurons, shows you that distinct pathways are organized on distinct dendritic domains. So to summarize this really complicated figure, if this is an RSE neuron, the visual inputs are coming onto this perisomatic region. The motor inputs are coming onto the proximal dendrite, and the thalamic inputs are predominantly concentrated on the apical dendritic tuft. If this is true, and we do see distinct pathways being organized on distinct dendritic domains, and if we think that only inputs that are going on overlapping dendritic branches can be linked, then this would indicate that only inputs that are similar in nature, for example, thalamic or motor, can be linked together. And this idea that inputs that are impinging on same dendritic compartments are the only inputs that can be linked is supported by modeling data. So with models, we can force where two memories are being forced. And if we make sure that only separate dendrite gets input for two different memories, as opposed to overlapping dendrites, we again see no memory linking. So indicating that this co-overlap or the pathways being impinged on the same dendritic domains is an essential factor for memories being linked together. And then finally, our lab and I would be very interested in knowing what the mechanisms of these compartmentalized dendritic plasticity are. We obviously were very agnostic to what mechanisms were in play when we were testing this idea, but now that we know that indeed there are compartmentalized dendritic plasticity mechanisms, it will be interesting to see what mediates it. With that, I want to thank everybody for coming. Thank you so much. And I want to thank our collaborators, especially Josh. A lot of this work was done in his lab, Wando Ho and Yura Poirazi. And I did not get a chance to talk about work that we've done with Daniel and Payman's lab, but this has been, at least for me, collaborating with these people is a wonderful part of being at UCLA. And in terms of our lab, I really want to acknowledge 3 undergrads, Sanana, Asla, and Irene. They were a joy to work with, and a lot of this work would not have been possible without them. I also want to acknowledge Ayal and Shan. They've always lent their expertise, and they've always been very helpful. And finally, I want to acknowledge Daniel. He has been instrumental in our lab's ability to be able to look at, how these neural calcium dynamics are affecting behavior, and really we've been able to dive really deep into that data. And last but not the least, Alcino. Thank you so much. I have had the pleasure of having a lot of independence and a lot of support in lab, and I think that's a rare research environment. And thank you for fostering it. I am very grateful. But I hope with that we have time for some questions.